All right, so this is the digestive system. It is found in chapter 22 of your textbook. And before we get into the anatomy and then a little bit into the physiology today, I want to show you this image here. This is essentially showing us how all of the different tissues and organ systems that we have started covering and will continue to cover are interacting with each other. And so here's the cardiovascular system. Remember that the cardiovascular system is literally like in your body. Once something is in the blood, it's going to be circulated throughout all of your different tissues, and therefore it is in your body, as opposed to the air we breathe, right, is isolated within the alveoli, and unless oxygen unless oxygen or whatever comes into our body, that stuff, that air, and as we're about to discuss, the food is invisible. We can't see it on the outside, so it's kind of hidden in our body, but it's not yet in our body until nutrients or oxygen, etc., cross over these epithelial membranes and enter into the blood. Of course, then the blood is going to distribute those substances throughout the body, delivering nutrients to our tissues, also delivering oxygen to our tissues. Okay, remember that our tissues, at the same time they are taking in food and they are taking in air, they are doing their thing, right? So skeletal muscles are generating ATP, they are, having sliding filaments, etc. And so in order to complete those functions on a daily basis, our tissues are also making waste. And so that waste will then be deposited within the blood, and it is eliminated by our urinary system. We're getting there. And of course, CO2, another metabolic byproduct, is eliminated via our respiratory system. Okay, so keep in mind that even though we are talking about each individual organ system independently of the others, they are actually working very closely together. Okay, so no organ system is independent of any other organ system. If for no other reason than the blood connects them all. Okay, so the digestive system is essentially one continuous tube all the way from mouth to anus. It is highly specialized, and essentially one of the main modifications or adaptations present within the digestive system, this long tube, is the need for an increase in surface area. Okay, so essentially we are going to see that even though the physical space between mouth and anus may only be like 3 feet, we're going to see that the digestive system is going to be very coiled, and therefore it provides a lot more of an opportunity for the functions inherent to the digestive system. That is, specifically, breaking down these larger molecules into small molecules that can actually be transported within the blood. Okay, and of course, there's a lot more to it than that, but that is the primary function of the digestive system. We can call this long continuous tube the alimentary canal. We can call it the gastrointestinal tract, gastro meaning stomach, intestinal meaning intestines, or we can simply call it the GI tract. Okay, so as you are studying and watching other videos, and even I will use all of these terms interchangeably. Okay, just like we always do, I am going to essentially follow the path of food from the oral cavity all the way until the anus. And so let's just very quickly walk through the different organs that are present within the alimentary canal. And so first we have the oral cavity. Food is swallowed through the pharynx. Right? We've talked about that a little bit with the respiratory system. With the swallowing reflex, food and drink is diverted down our esophagus, ultimately into our stomach, where it's kind of stored for a little bit, and little bits of this food mixed with stomach acid is going to then be passed from our stomach into our small intestine. The small intestine contains 3 kind of sub-organs or regions, the duodenum, next is the jejunum, and then finally is the ileum. The small intestine really is the main site of digestion and absorption. right? We'll see that all sorts of other things are happening throughout the entire tract, but the small intestine is really where the overwhelming majority of the magic is happening. And so the small intestine is the longest of all, essentially to provide more opportunities for digestion and absorption. From the small intestine, the food digested at this point, it is just about feces, is passed into the large intestine. The large intestine is divided into ascending, transverse, descending, and sigmoid colons. Make sure that you look at those digestive models in order to get a feel for which is which. And then finally, it is truly feces at this point. The food, feces, is now passed from the sigmoid colon into the rectum, and any waste is then released via the anus. Okay, so this entire tube houses ingested food and ultimately the waste products of said ingested food. Right, so that is the canal itself. But as we see in this diagram, there are a lot of extra structures that I haven't mentioned. These are called accessory digestive organs. That is, food is never going to directly pass through them, but the function of the digestive system is completely dependent upon these extra organs. And so the teeth and the tongue, the extrinsic salivary glands, the liver and the gallbladder, as well as the pancreas, are very important in the digestive system. Okay, and so before we elaborate on these different organs, I do want to remind you that there is a difference between different types of secretions, endocrine versus exocrine. We, of course, have talked about the endocrine system. These are organs or glands that release hormones into the blood, and that allows these ligands to, of course, circulate throughout the body and control organs that are possibly far away. And so endocrine means secretion into the blood. Exocrine means secretion into a duct, so usually onto the surface of something. So for example, our salivary glands are releasing saliva into our oral cavity. Okay, saliva would do us no good if we just secreted it right into the blood. So salivary glands are exocrine, and something like the pancreas secreting insulin would be endocrine. Okay, so we're actually going to start our discussion of the digestive system by talking about some of the accessory digestive organs. Again, the function of the digestive system is very much dependent on these accessory organs. And so I want to add in those details first, and then we're going to start in the oral cavity and go all the way down. Okay, so the first accessory organs I want to talk about are the liver and the gallbladder. The liver is responsible for producing, among many other things, about 1 liter of what is called bile every single day. Okay, and so we'll see how that works here soon, and we'll talk about the function of bile, but essentially bile is both a waste product as well as a something that helps us with digestion. So it's going to facilitate digestion. And so in the liver, we know that blood is passed up through the hepatic portal vein. Okay, and so blood is filtered, and in the process of filtering that blood, the liver makes bile. In order for the bile to go from the liver to essentially the small intestine to facilitate digestion, the bile is then passed through a series of ducts. Okay, there is a circular muscle, and circular muscles are going to become very important in this in this organ system. There is a circular muscle called the hepatopancreatic sphincter that is responsible for regulating how much of this bile, as well as some secretions from the pancreas, are going to end up in the duodenum. Okay, and so if we zoom in on this over here, what we can see is the wall of the duodenum. Okay, we can see the this green duct here coming from the liver and the gallbladder. We can also see a duct coming from the pancreas, and this circular muscle surrounds both of them. And so when the circular muscle relaxes, it's going to allow bile as well as pancreatic juices into the lumen, the opening of the duodenum or of the small intestine. Okay. Essentially, we want to store bile in the gallbladder unless we are eating. Okay, so bile is going to help us specifically to break down lipids. We'll get there. So we want to keep all of that, the lovely juice, that bile, in the gallbladder as a storage facility until we eat. Okay, so unless this hepatopancreatic sphincter is open, the bile is going to flow from the liver into the gallbladder. Okay, so from the liver down and up into the gallbladder here. Okay. We are going to see that this process of digestion within the intestines and the communication back and forth between the liver and the gallbladder is regulated by hormones that are produced in the intestines themselves. So we're going to see that the digestive system isn't just digesting food. It is also a powerful series of endocrine organs, and it has its own brain, right? It has its own nervous system, the enteric nervous system, embedded within the walls of the entire tract. Okay, so just something to keep in the back of your mind. Again, we're going to pull all of these pieces together as we go along, but as food goes from your stomach into your small intestine, into your duodenum, that is going to trigger the release of a hormone called CCK, cholecystokinin, which is going to open up that hepatopancreatic sphincter and allow bile to enter the duodenum. Okay, so again, the liver, the gallbladder, the pancreas are not physically digesting food. They're not physically interacting with it, but their secretions are going to facilitate the digestion. And they do that by controlling or by releasing these fluids into the duodenum when the hepatopancreatic sphincter is relaxed. Okay. Also, the walls of the gallbladder are muscular, and so we actually get a lot of bile released when we have lipid-rich food in our diet. I start off with that because this biliary tract historically has been a little bit challenging for students to understand. So I want to emphasize that learning the different ducts as well as the sphincters and et cetera are really important here. And so just one more time, I want to walk through here. The gallbladder releases bile through this duct right here called the cystic duct. Cystic means gallbladder. The liver is going to release bile via the common hepatic duct. Hepatic means liver. Common indicates that it's taking multiple ducts, right, the right and left, from the different sides of the liver. The cystic duct and common hepatic duct are going to join together to form the common bile duct. Okay, and the common bile duct is going to join together with this vessel right here, which is the main pancreatic duct, to form this kind of opening over here. Right, this is called the duodenal ampulla, which then can release both of these secretions into the duodenal lumen. In one of your laboratory models, I point out to you the duodenal papilla. Essentially, this is the junction of these two ducts, kind of sticking out just a little bit into the duodenal lumen, and we call it the duodenal papilla. Okay, so again, I start off with this so that you have the most time to learn these different ducts. Okay, so what else do these accessory organs do? Okay, let's start by talking about the liver. The liver is the largest gland in the body. We have already learned quite a bit about liver secretions. We've talked about proteins that are circulating within the blood that can be essentially turned on and lead to blood clotting. We've talked about other proteins that the liver makes that essentially transport iron, that can lead to blood pressure regulation, right? So the angiotensinogen is a liver protein, right? So the liver is secreting so much stuff. Also, it is the largest visceral organ in the body. if we were doing a dissection, you would see that the liver really does take up like half of the abdominal cavity. It is huge, especially in something like a cat that eats all sorts of kind of funky stuff and therefore needs a big liver in order to process all of that kind of funky stuff. Okay. The liver can be broken, the human liver that is, can be broken down into 4 different lobes. And again, the lab models will hopefully help you to see these a little bit more easily. But the right lobe is the largest. The left lobe is kind of triangular in shape. If we flip the liver over and look at the inferior aspect, so this is out towards the stomach, this is towards your back. We can see the right, we can see the left. Also, we can see this little square shaped lobe, that is the quadrate, right? So quad 4, so 4 sides to a square, and the caudate lobe here. Okay, the liver has a series of ligaments attached to it. First of all, the falciform ligament is going to separate the right and the left lobe. This here is the falciform ligament. Just kind of dividing the liver in 2. And essentially its job is to suspend the liver from the diaphragm and the anterior abdominal wall. So it's going to keep this huge organ kind of superior with, or not kind of superior, the most superior organ within the abdominal cavity, and it's going to keep it in place. All right, it's very heavy, so this is a very strong ligament. Okay. The falciform ligament directly leads into the round ligament, also called the ligamentum teres. This ligament is actually a leftover feature from your fetal development. So just like we saw with the heart, there are a few like scars left over from fetal vessels and fetal passageways from the right into the left atrium, etc. This actually used to be your umbilical vein. So going from your navel, your belly button, and actually delivering blood that has just picked up oxygen from mom directly to your heart. Okay, and so that structure never actually disappears. It's going to stay within your body. It just doesn't need to be a blood vessel anymore after you're born. And so it turns into a ligament. And so actually, this ligament is why sometimes getting a belly button piercing can be so very dangerous. Literally, your navel and your heart are still connected. So if you get an infection in a piercing or an umbilical hernia or something, that infection can travel up this ligament, get into your heart, and therefore be circulated throughout your entire body. Right? So that's, you know, it doesn't seem like a piercing could be that dangerous, but these particular piercings have been fatal in the past, and this ligament is the reason why. Okay. Another structure that I want to point out is called the lesser omentum. Remember that the serous membrane within your abdominal cavity is called the peritoneum. And it is essentially going to wrap over the surface of a lot of the different organs. In this case, the peritoneum goes over the anterior surface of your liver, it goes over the posterior surface of your liver, and it joins together. So we have two layers of the peritoneum before coming to the stomach, where it will then split again and go around the stomach. And so in between the liver and the stomach, there is this double layer of peritoneum that we call the lesser omentum. This double layer, of course, functions like any other serous membrane. It is going to hold your organs in place, specifically allowing the stomach to like hang off of the liver. Also, we can store a little bit of fat within this double membrane as well. Okay, so in this image here, we can see that the peritoneum wraps around the liver, then wraps around the stomach, and wraps around the liver to the other side. And here, in between, is the lesser omentum. Okay. In a future slide, we are going to see that we also have a greater omentum, and this one is pretty wild to see. I'll have to show you guys what that looks like, but omentum essentially means like lacy curtain, and so this is another double layer of peritoneum, which is filled with fat and essentially blankets over the entirety of your intestines. That's a pretty wild stuff there, to be continued. Okay. Final feature of the liver that I want to point out on this slide is that the hepatic artery and the portal vein enter the liver at the porta hepatis, right? So just like we saw in the lung, there is one location where everything enters and exits: blood, in this case, bile as well. Everything is going to pass into and out of the organ from the same spot. And this term essentially means the portal into the hepatic, into the liver. Okay. So the liver organization, let us transition from the gross anatomy into the microanatomy. The liver just looks to the naked eye like kind of flat, consistent tissue, but in fact, it is very highly organized inside. We have a lot of different blood circulations. We have bile circulation. So there's a lot of fluid flowing in all sorts of different directions. And so this video is going to break down what is flowing where and how, etc. Before we watch this clip, I want to point out that there is blood, right, entering the liver from the intestines, right? So we have nutrient-rich portal blood from the gastrointestinal tract that has just picked up all of the nutrients that we just spent so much effort digesting, absorbing, and now finally they're going to be transported to the liver before returning to the heart. So this blue vessel here is nutrient rich, but it is blue because it is oxygen poor. All right, so that blood has just dumped off a lot of oxygen to the intestines and in exchange picked up nutrients. So that is what the liver is actually going to be filtering. But also the liver needs its own oxygen supply. It needs its own blood, right? And so we have the hepatic artery, right, we talked about from the celiac trunk, that is also going to be entering into the liver from the inferior side, right? So the red one is going to feed the liver. The blue one is what the liver is actually processing. Okay. What exits the liver? Well, essentially, these blood supplies are going to flow through the liver and some magic liver stuff is going to happen. And the processed blood, now deoxygenated, is going to flow into the IVC, go back to the heart from the top of the liver. Okay, so the hepatic vein is going to drain into the inferior vena cava from the top of the organ. Right, so the blood supply is entering from the bottom and it's leaving from the top. Also, we have the drainage of bile. Right, so while the liver is processing this nutrient-rich oxygen-poor blood, it is making bile. And that bile is going to drain out the bottom. If we are eating lipid-rich substances at the moment, it's going to go right into the duodenum here. If not, it's going to be stored within the gallbladder. Okay, so let's take a look at how the liver is organized to facilitate these many different circulations.
Complex organ. One of the important functions of the liver is to receive and take up nutrients and other molecules absorbed from the intestines before the blood is returned to the heart via the inferior vena cava. The liver has dual blood supplies, one venous and one arterial. The portal vein, which carries blood from the spleen, pancreas, and intestines, contributes about 75% of the blood volume to the liver. This blood is rich in nutrients and other absorbed molecules, but relatively poor in oxygen content. The hepatic artery, a branch of the celiac trunk from the abdominal aorta, contributes about 25% of blood volume entering the liver. This blood is well oxygenated, but nutrient poor. Blood from the portal vein and hepatic artery mixes as it enters and passes through a liver lobule, the classical structural unit of the liver. Blood then percolates through the lobule and is collected by the hepatic or central vein. Central veins unite to form larger sublobular veins, which ultimately join the hepatic veins. Blood flows to the heart via the inferior vena cava. Bile comprises the exocrine secretory product of the liver. It is stored in the gallbladder. During digestion, bile is released from the gallbladder through the cystic duct and to the duodenum to aid in the digestion of fats. The filtration of blood and the production of bile takes place within the structural and functional unit of the liver, the lobule, where the terminal branches of the vessels carrying incoming and outgoing blood meet. The liver lobule is roughly hexagonal in cross-section. In the center of the lobule lies the central vein, which collects blood from the lobule and carries it to the hepatic veins. Located at the lobule periphery are branches of the vessel supplying incoming blood, the portal vein and the hepatic artery. Also located here is the bile duct. Collectively, the portal vein, hepatic artery, and the bile duct together comprise the portal triad. Taking a horizontal cross-section of the lobule gives us an arrangement typically seen in histological preparations. In this view, we can identify the central veins and associated portal triads. There are two prevailing views of liver lobule organization: the classic liver lobule, or anatomical model, and the liver acinus.
Okay, so we won't go into the histology, but I wanted you to see all of these different flowing substances throughout the liver. In the video, she mentions that the classical structural unit of the liver, even though it's not visible with the naked eye, is called a lobule. Okay. The lobules, as she said, are roughly hexagonal in shape. And so here we can see essentially the histology that she was just beginning to show us. And so let us focus in on one of these lobules and take a look at the blood supply as well as where the bile is going. Okay. At each of the corners of the hexagon, there are three different vessels. The three vessels collectively are called the portal triad. Okay. One of the vessels is called the bile duct. Right? And bile in this system is always going to be shown to us in green because it's green in real life. And so this bile duct is going to be receiving bile that is made by the liver cells. And so the liver cells, or hepatocytes, are these little brown guys here, and all of the other colored lines are the different vessels that we're talking about. So the bile duct is collecting bile that has been made by these hepatocytes, and it is going to drain the bile down and out of the liver. The second vessel is called the portal arteriole. Remember, arterioles are delivering oxygenated blood to a capillary bed. And so this is the incoming red oxygenated blood that has just come into the liver, essentially from the aorta. And so that is going to be the red vessel right here. This is feeding the liver itself. And finally, the hepatic venule. This is what is bringing us nutrient-rich, oxygen-poor blood from our digestive system. Okay, so we need to process all those nutrients in the liver before the blood is returned to the heart. And so this this blue right here, I know my colors, is the hepatic, or it's the portal or hepatic venule. And so this is a liver lobule, but from a slightly different perspective. We were just looking up here, right? So this is essentially the superior aspect. Now we are looking at the lateral aspect of the lobule. We can, of course, see the portal triad. Of course, the arteriole and the venule are both going to be delivering blood to this structure. The blood is essentially going to flow from the portal triad through these vessels. They are sinusoid capillaries, right, to refer back to what we've discussed before. And the blood from the triad is going to be drained up and out the central vein and ultimately to the IVC and the heart. Okay, at the same time, we have these hepatocytes creating bile and depositing that bile into what is called the bile canaliculi, or canaliculus for singular. And so these tiny little vessels are collecting bile all the way throughout, and essentially that bile is moving in the opposite direction to the portal triad and out towards the digestive system down here. Okay. Remember that these capillaries are sinusoid. They are the leakiest of all the capillaries in our body. They allow huge substances like red blood cells and huge proteins and such to cross over the capillary membrane and into the blood. Okay, so These capillaries, again, are super leaky. They are present between the liver cells, so these hepatocytes. Okay. Also present within these capillaries associated with the liver tissue are hepatic macrophages. So hepatic for liver, macrophages for big eaters. And so these are essentially cells whose job is to engulf, right? Kind of like Pac-Man, engulf anything that is very large and doesn't look quite right. So this can be red blood cells that are starting to break down because they don't have nuclei to be able to fix themselves. They can be removed by the liver, as we've discussed before. This can be bacteria that have gotten into your body and are trying to infect your tissues. These cells are going to get them out of the way and break them down so they're no longer a threat. And so on and so forth. So anything that doesn't look quite right, we can see these Kupffer cells, these hepatic macrophages that are essentially patrolling the blood coming up from the intestines and coming into the liver from the aorta. Okay. The most important cells in this entire system, this entire process, of course, are the liver cells themselves. The liver cells, which are here in like tan or light white here, these are called hepatocytes, cyte for cell, hepato for liver. So what exactly do they do? This is a very abbreviated list. We really are talking about hepatocyte functions throughout this entire course, but I just want to summarize a few of them for you now. First of all, they process bloodborne nutrients, right? So the nutrients that we just digested and absorbed within the intestines are now going to be processed here. And shortly, we're going to talk about glucose metabolism and essentially how it's the liver's job to regulate how much glucose and other nutrients are stored and how much fat we're making, et cetera, versus how much is circulating in the body. So really it is the liver's job to make sure that the entire body is fed. And it's actually, it's pretty selfless in its process too. It actually sacrifices its own nutrients for the rest of the body. But again, we'll get into that here in a second. It is the hepatocyte's job, the liver's job, to store fat-soluble vitamins. We have 4 different types: vitamin A, D, E, and K. When we eat them today, our body, our liver, actually stores them, the little vesicles within the hepatocytes, so that if we don't eat, vitamin A a month from now, we can dip into these reserves and therefore supply the needs of the body, right? So even vitamin K, which of course we know is really important to blood clotting. And so even if we don't eat vitamin K for the next month, we can still make those clotting factors and still generate a blood clot in a good way. The function that you're probably most familiar with is detoxification, right? So of course your liver has to work really hard when you ingest kind of extracurricular substances like alcohol, but also any other kind of toxin that maybe you're not even purposely ingesting. You may not even be aware that you're ingesting. We actually absorb a lot of chemicals from our environment through our skin and through our, of course, delicate respiratory membranes. And so the liver really does a great job at breaking those substances down. Okay. Also, hepatocytes produce, give or take, a liter of bile every single day, which, as I mentioned, is critically important in digestion. Okay. Also, I want to throw it out there that the liver is the only organ within your body that can regenerate, right? So these cells actually have the ability to make more of themselves. So if you donate part of your liver, right, when you're alive, it will replace itself, right? So there's no other organ in the body that can do that. If you lose an eye, if you lose a muscle, if you lose part of your heart to myocardial infarction, right? That tissue's gone, right? There's no way you can get it back. The liver is the only one that can grow back if you lose part of it. Okay. To elaborate a little bit more on how the liver feeds the entire body. I have borrowed this from a peer-reviewed paper, but I think it does a great job at kind of summarizing the different metabolic states of the body and how the liver is responsible for making sure that we have enough nutrients to go to our tissues, even when we're not eating right now. Okay, so the liver, of course, stores fuel, right? Stores nutrients when food is abundant, when we're eating. So it can take it out of the blood and store it within its cells. But then when other tissues start to need that fuel, it can mobilize it and feed those tissues. There are a couple different types of molecules that the liver produces and works with to make sure that the body is fed all the time. And they're kind of broken down in this nice chart here. So first of all, when we are fed, right? So when we are eating breakfast or for the next hour or so after we're eating breakfast, our body is fed, right? So our digestive system is breaking down those polymers, and we are absorbing nutrients into our blood. That blood, of course, circulates throughout the body and feeds our tissues directly. And so things like glucose can go directly from our digestive system to our tissues. And so we utilize these dietary carbohydrates when we're eating or soon thereafter. The time between meals, right? So you ate breakfast this morning before class, and then if you don't eat again until 1 or 2 in the afternoon, that is a short-term period of fasting. Okay, and so that those dietary carbohydrates, that glucose circulating in your blood, is not going to be sufficient to sustain your body's activities for that many hours. And so our liver actually can start breaking down polysaccharide molecules called glycogen, can start pulling off glucose, dumping it into the blood via those sinusoid capillaries, and therefore it can pretty much get the body through those periods of short-term fasting. Okay, so we are constantly going back and forth between the sugar we eat and the sugar we've stored, and the liver really is responsible for regulating those processes, making sure everything in the body has enough to eat. Okay. Long-term fasting, right? So if you go until late tonight or to tomorrow or even longer than that, this becomes long-term fasting. Essentially, the liver can break down all of its ready supply of glycogen, therefore feeding the rest of the body. But when we run out of those stores, the liver has to kick into kind of plan B, right? So essentially, the liver can start to break down other substances, such as proteins, right? It can start to break down fats and create what are called ketone bodies, right? So if your body is in such a dire situation where you haven't eaten in so long that you've burned through all of your dietary sugar, all of your stored glycogen, your body is going to start kind of prioritizing what needs to be fed. And so your liver, in response, makes something called ketone bodies. And these ketone bodies are, of course, a ready source of energy, but they're really only able to be used by a couple different organs, specifically the brain and the heart, right? So like the most important organs of your body. One is controlling the whole body, one is delivering blood to the whole body. And so those are what are most important to keep us alive. And therefore, those are the ones that can use this kind of emergency fuel, right? You have exhausted all of your other stores, and now you need these ketone bodies, but only certain organs can use them. And so the liver kind of makes that decision, and it says, you know, the brain and the heart, we need to keep them going, but everything else, including the liver, so again, it's very selfless here, the liver can't even use these ketone bodies. And so, again, the liver is really important in making these decisions, keeping the entire body going, even though it cannot use some of those energy sources. We're going to come back to ketone bodies later this semester. Ketone bodies are actually pretty acidic. And so if you have an issue with glucose metabolism, such as if you are diabetic, your body or your liver is producing a lot more of these ketone bodies, which changes your pH and can lead to some pretty severe side effects. Right, so ketoacidosis. Right, we'll come back to that. Okay, so let us zoom out again and look at the liver lobule. This is just a slightly different perspective. I want to walk you through what the liver is doing step by step. So first of all, blood enters into the liver sinusoids. So these right here, from the hepatic portal vein, as we know, as well as the hepatic artery. So from this and from this, blood starts to flow towards the central vein. So from the corners of the lobule towards the middle. Okay. As the blood is flowing through these sinusoids, these hepatocytes here are able to take out extra nutrients. All right, so maybe we just ate a meal and so our blood is filled with sugar, and so we are going to bring that extra sugar out of the blood and store it as glycogen, for example. Anything that is toxic, we will pull out of the blood as well. Anything that is a little bit funny, doesn't look exactly right, are going to be broken down by these Kupffer cells. Okay, so the blood is going to be modified quite substantially by the cells as they flow through the sinusoids towards the central vein. And the central vein is going to collect blood from the lobules and then ultimately empty it out up into the inferior vena cava. While the blood is moving from the corners to the central vein, bile is going to be deposited into these bile canaliculi by the hepatocytes. Okay, so the bile is going to flow out towards the corners and again down to be stored in the gallbladder or to be deposited directly into the duodenum. Okay, so really, what is bile? I keep talking about it. I mentioned this, I think like the first day of class. What is bile? All right, so it's this yellowish green substance. It is alkaline, which is going to be important to digestion here soon. And it contains a variety of things produced by the liver, or at least processed by the liver, such as bile salts. Bile salts are derivatives of cholesterol. So these are hydrophobic substances that play a role in emulsifying the lipids within your digestive tract. So essentially you can think of these bile salts as like Dawn dish detergent, right? So when you wash a greasy pan with just a sponge, that grease just like sticks together and of course it repels the water, but then you add the dish soap, and that dish soap actually breaks those lipid molecules down into smaller little pieces so that you can wash them away and they're no longer repelled by water. And so that's exactly what these bile salts are doing in your digestive tract. They're going to take big globby lipid droplets and they're going to make them much smaller so that you can actually digest them and take them into your blood. Okay. Bile also contains excess cholesterol. Okay, it contains phospholipids, which are also going to be important in this emulsification process. It contains excess electrolytes. Okay, and what we've talked about since the first day of class, it contains bilirubin. Okay, so bilirubin is what gives it its color. We know that bilirubin is the byproduct of breaking down hemoglobin. Okay, and so this breakdown of hemoglobin is going to occur in the spleen. It's going to occur within the liver as well. And so this bilirubin has to get out of your body somehow. It is going to be dumped into the intestines. And so a lot of that bilirubin ends up in your stool. And so that's what gives your stool its brownish color. But then some is actually taken back up into the blood and exits via the kidney. So essentially we need to get rid of this byproduct of our red blood cell breakdown, and we do that via our urine and our feces. Bile is made, right, constantly by the liver, about a liter every single day, but it's kind of expensive to make. Okay, so all of those different components all have a different role in digestion, and so we actually recycle a lot of that bile, right? So it's not all going out in the feces and the urine. Instead, it is largely recycled. So we can keep on using that dish soap over and over and over again. We can keep on using cholesterol, which of course is really important to all sorts of things, over and over and over again. And so what this little sketch is showing us is that the liver makes the bile goes into the duodenum, and over the length of the small intestine, we actually absorb a lot of those different substances, particularly the bile salts, which are important in emulsification. Okay, so we reabsorb those substances into the blood of the intestines, and of course we know the intestinal blood goes back up into the liver via the hepatic portal vein. Okay, so then the liver would pick out those bile salts from the blood, dump them back into the bile ducts, and we recycle them over and over and over again. As I said, the bile is going to flow from the liver directly into the duodenum if that hepatopancreatic sphincter is open. Right? If we are actively eating and digesting lipid-rich food at that moment, it's going to go right into the intestines. If not, the bile, right, a liter of it, is going to be stored in the gallbladder. Okay, and so the gallbladder again is on the inferior aspect of the liver, and the bile can travel up the cystic duct to be stored in this floppy, muscular lined sac. Okay. The gallbladder's job, of course, is to store the bile, right? If we're not eating lipid-rich food right now, it stores the bile, and also it concentrates the bile. So the longer that the bile is sitting in the gallbladder, the more water is going to be reabsorbed from it, and therefore it gets more and more and more concentrated. The gallbladder also has a role in lipid digestion in the fact that it is responsive to hormones released by the duodenum that control when and how much bile is released into the duodenum. Okay, which leads me to talk very briefly about gallstones. Maybe you know someone who has had gallstones, maybe you have had them yourself. If so, I'm really sorry to hear that. So gallstones are essentially these hardened substances in your gallbladder. Okay, there's a couple of different types. Most of the gallstones that are produced in America are cholesterol stones. These are the yellowish green ones. And essentially this is, or this happens when you have very high cholesterol in your blood, right? And so your liver is constantly pulling that cholesterol out, dumping it into the bile. And so you have a super thick bile filled with cholesterol. And so sometimes that cholesterol can stick together and make these kind of squishier yellow-green stones. Okay. The other type of stone are the bilirubin stones. So these are pigment stones. They are darker in color. And so this can happen when you are breaking down way too much blood, right? So more blood than you normally would be. And so you would be therefore making lots of bilirubin, and that bilirubin can stick together in your gallbladder and make these darker, like blackish color stones. Also, if you have insufficient bile salts, if you have inadequate emptying of your gallbladder, right? So if there's some kind of blockage or something that is preventing the bile from going from the gallbladder down into your intestines, essentially your gallbladder is going to continue dehydrating or concentrating that bile until it becomes little rocks, right? So it's so dehydrated that it is solid instead of liquid. Another thing that can cause gallstones is cholesterol medication, right? So if you're trying to treat your high LDL, your high bad cholesterol, your liver, of course, pulls more of that cholesterol out of your blood, adds it to your bile, but of course, too much cholesterol sticks together and forms those yellow-green ones. Also, a poor diet. So if you are eating so much fatty food, you can get gallstones because of that extra fatty substances, those extra fatty substances that end up in your bile. Okay, let's see. Good, we can start with the pancreas. So the liver and the gallbladder are both these accessory digestive organs. They never physically interact with the food, but they are very important in particularly digesting lipids and in processing all of the nutrients that have just been digested by that alimentary canal. The other accessory digestive organ that I wanted to mention today is the pancreas. We've seen the pancreas when we talked about the endocrine system, but now let's really get into what it does. The pancreas is inferior to the stomach. It has both endocrine and exocrine function. So it's both producing hormones to be dumped into the blood, and it's producing digestive enzymes and buffers to go into the duodenum. Okay, so let's talk a little bit about the exocrine function first. The pancreas, it has specialized cells called acinar cells, and these, when stimulated, are going to produce an alkaline fluid called pancreatic juice. Keep in mind that when the food, or the chyme as we're going to call it, passes from the stomach into the duodenum, it has a pH of like 2. Okay, sorry for that. Has a pH of like 2. And when that really acidic fluid gets into your intestines, it can burn the lining of your intestines. And so we add this alkaline rich buffer juice to that chyme, to that food, to immediately bring the pH up to like 8. Okay, and so the pancreas is one of the ways that we do that. Okay, and so these acinar cells would produce those buffers, dump them into the pancreatic duct, which would then, as we can see up here, join together with the bile duct to dump those fluids into the duodenum via that major duodenal papilla, this little dot right here. Okay. Also, these cells are responsible for producing enzymes, right? So the pancreas isn't digesting anything, but it is the only organ in your body that can produce enzymes for every single biomolecule type, or biomolecule class. So the carbohydrates, the proteins, the lipids, and the nucleic acids all are digested by enzymes from the pancreas, not from the small intestine, not from the stomach, not from the mouth, the pancreas. The endocrine function. The pancreas also produces several types of hormones, and these are going to be produced by a different type of cell within the pancreas. All right, so here we can see the acinar cells depositing their juices into the small intestine. Here we can see a pancreatic islet. Okay, it does not have a duct associated with it, but it does have a blood vessel associated with it. And so that should indicate to us that these pancreatic islets, or islets of Langerhans, are endocrine in function. Okay, first of all, the pancreatic islets produce insulin, okay, produced by the beta cells of the islets, as well as glucagon. Okay, so insulin is generally released. Well, sorry, one second. There's a third type of hormone, and that is somatostatin. We're not really going to talk about this very much, but it is another pancreatic hormone. Okay. Insulin and glucagon are essentially going to be released to inform the liver, inform the tissues of essentially what we should do with glucose. And so I bring up this image right here to show you that glucose is what is absorbed into the blood after digestion. Glucose can be utilized by our tissues to create ATP. So here is glycolysis making pyruvate. Pyruvate can then be used by the mitochondria to make lots and lots of ATP, right? So we can either directly use the glucose in our tissues or we can store it. Okay, so glucose can be stored as glycogen, right? This polysaccharide molecule. And the process of taking glucose and storing it as glycogen is called glycogenesis. We are literally generating glycogen. Okay. If there is not enough glucose in the blood, right? Glucose is gone. Glycogen can then be broken down into glucose. Okay, so that process is called glycogenolysis. So the lysis indicates the breaking down. So lysis here takes this storage molecule and produces usable glucose from it. So now our tissues would be able to have enough to eat. We can also, particularly in the liver, make new glycogen, make new glucose. So we can take proteins, we can take fats, and we can make new neoglucose from those substances. Okay, and so again, I bring this up now because insulin and glucagon are essentially going to push this reaction in one way or another. Okay, so insulin is released when there is a lot of glucose in the blood, or there is about to be a lot of glucose in the blood. Okay, so this is going to stimulate this reaction to go to the left here. So essentially, insulin, when detected by those hepatocytes, is going to increase how much glycogen is stored. And it is going to decrease how much glycogen is broken down and how much new glucose we are making. Okay, so essentially, it is pushing everything in this direction. On the other hand, the glucagon is released when glucose is gone. Okay, and when detected by the hepatocyte cells, glucagon is going to trigger the liver cells to start making new glucose, to start breaking down glycogen, and to stop storing so much glycogen. Okay, so ultimately, when glucagon is detected by the liver, it is going to increase glucose production and release into those sinusoid capillaries. When insulin is detected, it is going to increase glucose uptake and storage. So we're going to save that energy for a later date. We're going to end today with this image. Lots of different things going on, but it is really important to understand this glucose metabolism. Fed state, you just ate, right? Insulin detected by the liver cells is going to stimulate glycogen synthesis. So here we can see glucose coming into the liver, and it is therefore added to existing glycogen molecules. Okay. On the other hand, glucagon is released when you are fasting or even when you're exercising, right? So there's not enough glucose in your blood, glucose is gone. And so glucagon is going to trigger more new glucose creation, right? So we take glycerol, lactate, amino acids, all these different types of molecules, and we can literally make new glucose to be released into the blood. Also, glucagon directly leads to glycogenolysis, so taking this storage molecule in the liver and releasing glucose from it. Okay, also, when you are fasted or when you are exercising, you also release a little bit of epinephrine, which also contributes to these processes. Okay, this is a summary of the exocrine structures of the pancreas and the endocrine structures of the pancreas, which release these hormones to ultimately inform the liver what to do with sugar.